Okay, so hello everybody. So thanks a lot for joining for this seminar. So we are going to start. So just a few technical points before we start with the seminar of today. So for those on Zoom, as usual, you can report any technical issue in the chat, and you can ask your question via the Q&A tab, and we will be able to let you speak at the end of the talk and interact with the speaker if you want to ask questions or if you have any comments. And so with that, thanks very much both for coming, and I let it to Delphine to introduce today's speaker.
Thank you very much. Can you hear me well? It's fine. Yeah, so it's my great pleasure to welcome you. Eric Gangloff is from the University of Ohio, Wesleyan University. I don't know how to pronounce that, sorry for that. Eric is working on the ecology and on the physiology of reptiles, and he'll be talking today about lizards that have become invasive in Ohio and how they adapt to climate change too. And thank you so much for being here and for presenting your data, Eric.
Thank you very much, Delphine. Thank you to everyone for being here this morning, and a special thanks to Christophe Pruneau for inviting me to give a talk and for organizing a fantastic visit to Perpignan and to Montpellier this week. My name is Eric Gangloff. I'm here as a LabX tulip visiting scientist for the summer. My home university is Ohio Wesleyan University in the United States. And my talk today is titled Try and Try Again: Temperature-Oxygen Interactions Shape Performance Curves and Thermal Limits in Montane Ectotherms. And I'd like to begin with a narrative outline of what I'll present today. Chapter 1 is titled The World is Changing, and This Will Affect Life. This is probably no surprise to any of you. Chapter 2 is We Think We Might Understand How. Chapter 3, But We Don't. Chapter 4, So, being good scientists, we try something else. Chapter 5, and maybe this new thing is right. And Chapter 6, but probably not always, but that's okay because it keeps things interesting and gives us biologists jobs. So let's begin with Chapter 1, The World is Changing and This Will Affect Life. I'd like to begin with a series of images showing you a boat marina on Eagle Lake, which is a mountain lake in the Sierra Nevada mountains of Northern California in the USA. And this is the site of my PhD dissertation work, and also the series of images that I began my PhD dissertation defense with a few years back. And you can see back in 1979, this is an active boat marina. It's a very popular spot for people to vacation and go fishing. And if we fast forward to 2006, you can see it looks kind of the same, although the water level has dropped along the break wall there. By 2009, that water level has continued to drop, and you can see there's no boats now in the marina. By 2012, the water is so low that some of the larger rocks are emerging from the surface. You can see a kind of makeshift boat slip out by the mouth of the arena where the water is still deep enough for boats. By 2013, some grasses have started to move in, and the water is restricted to just some of the deeper areas. By 2014, that boat slip is now entirely on land, and there's just a few muddy puddles. And by 2016, the year I finished my PhD dissertation work, you can see what was once an active boat marina is now a grassy meadow around the lakeshore. Now, I present this series of images as just one example. All of you know in your different study systems and different ecosystems that you study how climates and environments are changing pretty rapidly. I like this example in particular because it illustrates the different ways in which habitat is changing. First of all, you can see dramatic shift to the structural habitat, which was what was once part of a lake, is now a grassy meadow. And this is due, at least in part, to human changes of the climate and climate warming specifically, which has caused this lake level to lower. So our job now as biologists is to understand how this change in environments will affect the organisms that call these places home. And to do that, we have a pretty good idea of how that might work in terms of temperature, which brings us to Chapter 2. We think we might understand how changing environments affect organisms. And to describe the effect of temperature on organisms, I'm going to show you a thermal performance curve. So this is a tool that's been used for many decades to describe the dependence of organismal performance on temperature. So on the x-axis here, we see a range of body temperatures, and the y-axis, some measure of organismal performance. Now, this can be performance at a subcellular level, for example, the activity rates of enzymes. It can be whole organism metabolism, or sprinting or running performance, locomotive performance. Or for microorganisms, for example, it could be something at the population level, like population growth. And this curve describes the temperature dependency of this performance across a range of temperatures. And from this curve, we can extract some important parameters. This includes the optimum temperature for performance. So this is the temperature at which that performance is maximized. And also the critical thermal minimum and critical thermal maximum. So these are the lower and upper temperatures at which performance becomes effectively zero and an organism loses functionality. We can also describe a performance breadth. So this is a breadth of temperatures across which an organism can attain some relative proportion of its maximal performance. And these curves have proved to be useful in a variety of contexts over many years. But to use these curves predictably, we need a mechanistic understanding of what causes this change in shape of the curve across these temperatures. And for lower temperatures, we generally understand this. It's due to thermal kinetics. In other words, as temperatures increase across this range of temperatures up to the thermal optimum, molecules are vibrating faster, interaction rates increase, and so organisms are able to do things more quickly. At the upper end of the thermal performance curve, however, we often, in many contexts, lack a solid mechanistic understanding of what drives this rapid decrease in performance at high temperatures above the optimum. To explain that, there's been a few competing hypotheses proposed in the literature over recent decades. The first hypothesis I'll show you here on the left, this is the classic hypothesis that postulates that it is changes at the subcellular level that drive this decrease in performance at high temperatures. So for example, proteins lose their ability to interact efficiently, they can denature, membranes can lose their fluidity and their integrity, and that can cause problems for these cellular processes, and that ultimately scales up and leads to failure at the organismal level. In the past 15 or 20 years, however, a more recent hypothesis poses that the problem at high temperatures is actually at a higher level of organization at the organ system level, and specifically that oxygen becomes limited at these high temperatures. So in this case, the increased temperatures increase the oxygen demand of cells, but the body is not able to deliver this oxygen in sufficient quantity, so you have a mismatch between oxygen demand and supply, and this leads to organismal failure at high temperatures. Now, this was a hypothesis proposed rather recently, and the work to describe this was primarily based in aquatic ectotherms. Now, at the time when I was a graduate student, I read about this and I thought it was a very exciting hypothesis, and there were relatively few tests of this in terrestrial ectotherms. So I thought, well, we'll design an experiment and we'll test to see if this is the case in a terrestrial ectotherm. And this is very exciting because as a PhD student, I thought, oh, I can demonstrate this, that this mechanism is important in this new taxa. This will be the first time that this is shown. This is going to be great. We're going to design an experiment to do this. And so we did. And that brings us to chapter 3, titled, But We Don't. So you can see where this is going already. Nonetheless, I want to tell you about this experiment that we conducted. And for this, I will turn to the Western terrestrial garter snake, Thamnophis elegans. This is a small, harmless, rather conspicuous species of snake that's found across much of Western North America. You can see the range map in purple here. And specifically, my PhD work studied populations of these snakes around Eagle Lake in Northern California, which is right around here. This is the series of images with which I began showing you the marina being converted to a grassy meadow. So within these well-studied populations of snakes, we designed an experiment to test this oxygen limitation idea. And to do this, we measured a number of physiological traits of snakes across a range of temperatures that represents their normal activity range, right up through temperatures that are just below what would kill them under this acute exposure. So we really wanted to push them to the edge of their limits, but of course not injure the snake in the long term. Our primary goal was to test this oxygen limitation hypothesis, one of the first tests in a terrestrial tetrapod. but we're also interested in generally characterizing the whole organism hormonal and energetic response to these high temperatures. And in other words, what's the physiological response of these snakes to these extreme high temperatures? And to do this, we exposed snakes to 2 hours at each of these temperatures. During this time, we measured their oxygen consumption, which serves as a proxy for their metabolic rate. We also collected blood samples in which we measured glucose, which is an important energy substrate, just as it is in humans. We measured the hormone corticosterone, or CORT, which in snakes serves a similar function to cortisol in humans. So it's a hormone that's commonly implicated in the stress response, but also serves an important role in maintaining energy balance more generally. And finally, we measured the hormone insulin, which plays a similar role in terrestrial ectotherms as it does in humans in regulating glucose levels and energy balance more generally. So here's what we found in our experiment. First, in terms of the stress response, this figure shows you the range of temperatures that we measured on the x-axis. The y-axis shows the concentration of corticosterone. And you can see here very clearly that these high temperatures represent a stressor for these snakes. The corticosterone stays relatively stable across the low temperatures, but when we get to the high temperatures, it increases rather dramatically. And we saw a similar response in glucose as well. This figure again shows your range of temperatures, our range of temperatures on the x-axis and the level of glucose in the blood on the y-axis. And we see a similar response here where glucose, circulating glucose is increasing at these high temperatures, presumably to mobilize energy stores to combat these high temperatures, either physiologically or behaviorally. For insulin, which is important for any energy regulation, we found an interesting result where insulin increased across the range of temperatures, but in a different, more linear pattern. And this suggests that insulin is playing a role in heat stress and in the response to heat, independent of its role in regulating glucose levels. And this was, in fact, the first time this was shown in an ectothermic vertebrate. So we were pretty excited about this result, and it pointed the direction to some new and interesting experiments understanding the role of insulin in reptiles and other ectothermic vertebrates. But finally, the result that would ultimately test this oxygen limitation hypothesis was the level of oxygen consumption. And you can see here, oxygen consumption on the y-axis and our range of temperatures on the x-axis, that a metabolic rate increases rather gradually with temperature up until the highest temperature, at which that metabolic rate increases rather dramatically. And this suggests that even at those temperatures that are just below the lethal limit for these organisms, they're not suffering from any kind of oxygen limitation. And so we found no support for this oxygen limitation hypothesis in snakes, suggesting that other mechanisms are responsible for organismal failure at high temperatures. So even though the experiment didn't support the hypothesis that as a graduate student I was so excited about, we did find some interesting results in that these extreme temperatures induce this hormonal and energetic response, and that these responses, importantly, are outside of the normal homeostatic range of these biomarkers. So this suggests that even acute exposure to these high temperatures can have long-term implications for the physiology of these organisms. So for example, under conditions of a heat wave, even under acute exposure, even if the animals can survive, there can be long-term consequences even after that short exposure to high temperatures. So as a graduate student, I have to say I was a little bit disappointed by this result. I had in my mind that I would find support for this hypothesis, and I didn't. But of course, being a good biologist, I didn't give up. Instead, I went back to the drawing board and we tried something else. And I want to tell you about this, what we tried next. And to do so, I'll turn back to the thermal performance curve. So here's another example of a thermal performance curve. We have this range of body temperatures on the x-axis again, and some measure of organismal performance on the y-axis. Now we know that thermal performance curves can change under a variety of conditions or ecological contexts. For example, thermal performance curves can change across an individual's ontogeny, or they can change in relation to other limiting abiotic factors. For example, salinity in a marine environment or oxygen availability that I'll talk about here in a minute. And these curves can change shape in a number of different ways. For example, curves can shift entirely to the left or to the right, which represents adaptation to warmer or cooler temperatures. A curve can change shape without the critical thermal minimum and maximum changing position, or curves can become narrower or wider, representing a kind of thermal generalization versus thermal specialist. So to understand how a curve will change shape under different conditions, we need to understand what's driving that shape in the first place. And to do that, my friend and colleague Rory Talamico and I proposed a new idea. And we looked at these two different hypotheses for how high temperatures might lead to organismal failure. And I mentioned that this was recently proposed in the literature and was actually quite controversial. There was a series of back and forth papers in the Journal of Experimental Biology. My understanding was that there was even some yelling at a meeting a few years back on camps that were in favor of this hypothesis and those that were opposed to it. So it was a rather controversial area. But we looked at this and said, well, why do these things have to be exclusive? There's no reason why different mechanisms can't be important under different ecological conditions. So we looked back in the literature on reptiles and we found support for this idea of how both subcellular components and organ systems could be important in different ecological contexts. And we proposed, with these ideas, we proposed the hierarchical mechanisms of thermal limitation hypothesis back in 2018. So I'm going to walk you through how this works. What you see here is a figure that shows a range of temperatures on the x-axis and oxygen demand and capacity on the y-axis. And you see two lines here. The blue line represents the resting metabolic demand of an organism. So this is the basal metabolic demand to meet just baseline functions to stay alive. The red line represents the maximum aerobic capacity of an organism. Now, these lines are fitted published data from an experiment in pythons in which they measured the metabolic rate of resting pythons that were fasted and also pythons that had just eaten a very large meal, which ramps up their metabolic rate quite dramatically, as you can see here. Now, what's important seeing these two lines representing resting and maximal metabolic rate is the difference between those lines, which is known as the aerobic scope for activity. This aerobic scope is essentially the capacity an organism has to increase its metabolism to perform important functions. And importantly, many of the functions that an organism performs that are closely tied to fitness are fueled by this aerobic scope. So for example, things like digestion, growth, and reproduction are all long-term functions that are fueled by this aerobic capacity of an organism. So this aerobic scope can be used as a kind of indirect proxy for fitness. So if we turn back to our curves here, you'll also notice that at very high temperatures, far out here on the right, these blue and red lines intersect at the aerobic critical thermal maximum, or aerobic T-crit. This is the temperature at which an organism's resting demand meets its maximum capacity, and the aerobic scope becomes zero. In other words, this is the high temperature at which an organism can't perform any functions other than meeting its basic metabolic needs and surviving. You'll also notice on this figure this vertical black line that represents the subcellular thermal critical maximum, or subcellular T-crit. This is the temperature at which those subcellular components, the proteins and membranes, fail, leading to organismal failure. And you'll see in this case that subcellular T-crit happens at a lower temperature than the aerobic T-crit. So we propose that whichever T-crit is reached at a lower temperature is the one that will be important in a given ecological context, hence the name hierarchical mechanisms of thermal limitation. Now, everything I've shown so far assumes an organism under conditions of normoxia, where oxygen is readily available. If we move organisms to conditions of hypoxia, where oxygen is limited, we can expect some predictable changes in the shapes of these curves. Specifically, this aerobic scope for activity will be reduced, and this aerobic T-crit shifts to a lower temperature and shifts to the left. Now, you'll notice here that this temperature is still occurs at a higher temperature than the subcellular T-crit. And if we translate this to a thermal performance curve for aerobic scope, we get something that looks like this. Under condition of normoxia, we have our thermal performance curve. You're all experts in thermal performance curves at this point. And if we expose organisms to mild hypoxia, we expect the shape to change predictably so that maximum performance is limited and this optimal temperature is downshifted slightly to the left. But under these conditions, there's no change in the critical thermal maximum. That critical thermal maximum stays the same because it's being driven by that subcellular T-crit. Now, if we move an organism to conditions of more extreme hypoxia, something like this, we again find that this shape changes in predictable ways. This aerobic scope for activity now is much more dramatically reduced across that entire range of temperatures. And now this intersection of the resting and maximum metabolic rates is downshifted to the left to a point cooler at a lower temperature than the subcellular T-crit. So bringing this back to our thermal performance curve, under extreme hypoxia, we find that maximum performance is dramatically reduced, that optimal temperature is further shifted downward to a cooler temperature, and under certain conditions, that critical thermal maximum can be reduced as well. So we proposed this new idea. We found support in this by looking back in the last several decades of literature and physiology in reptiles and amphibians. And we proposed this idea, and we're excited because it provides some very specific predictions, specifically that the thermal performance curves, they will change shape when oxygen is limited. And this can happen under a variety of natural conditions. So for example, at high elevations, in water, where bodies of water where oxygen becomes limited, in embryos developing in eggs, where oxygen diffusion is limited across the eggshell. So there's a variety of important ecological conditions in which oxygen is limited, and we wanted to understand how that might affect the shape of these thermal performance curves for different traits. So we provided these predictions, and specifically, or additionally, also that critical thermal maximum would be the most robust component. So a lot of research was focused on conditions under which this critical thermal maximum might change. Well, we wanted to say, even if the critical thermal maximum doesn't change, other changes in the shape of that thermal performance curve can be very important for organismal function and ultimately fitness. So we proposed this hypothesis. And of course, when you propose a hypothesis, the next thing you need to do is to go out and test it. And that brings us to chapter 5, titled, "And Maybe It's Right," which is an encouraging title. I'm going to talk here about a few experiments I conducted as part of Project Podarcis. Podarcis, this was my 2-year postdoc position with the CNRS in Moulis in France. And Podarcis is, of course, the genus name of the beautiful lizard that you see here. And also, I'm going to argue, is the world's greatest acronym, standing for Potential Oxygen Limitation of Distributions and Responses to Changing Climates in Ectotherms. So this was my postdoc position. And our idea here was to use the common wall lizard as a model organism to understand the responses of ectotherms to changing climates in montane environments. And the common wall lizard lives up to its name across its broad geographic range, covering much of southern Europe you see here. It's very common and conspicuous. You probably see them all around here. And they do, in fact, love walls and other anthropogenic structures. Now, across this broad geographic range, they inhabit a variety of different environments, but generally they are found at lower elevations. Now, in recent years, in the Pyrenees Mountains in the south of France, this species has been observed moving upslope, and moving upslope rather rapidly as temperatures have warmed. So we conducted a series of experiments to understand what the implications were as higher elevation habitat became thermally suitable, what might limit or facilitate the colonization of that habitat. And specifically, we were interested in how oxygen limitation at high elevations might limit the physiology and ultimately the colonization potential of these lizards. And so I want to describe some of the results of those experiments, but first I want to take a brief aside, if I could. I'm just going to grab a sip of water here. I'd like to take a quick minute to discuss my favorite short story by the author Herman Melville. That's the short story titled Bartleby the Scrivener, written in 1853. Now, this is a story, I'll make the connection with lizards clear in just a moment here. This is a story about a clerk working in a law office. And back in the day, the clerks had to copy all of the scripts of the law books by hand. It was a very tedious kind of work. And the story is about a scrivener named Bartleby who comes to work one day and his boss asks him to copy something and he refuses to do it. And there's a great quote in the book here when the lawyer who's in charge asks him to do the work, and he says, "Imagine my surprise, nay, my consternation, when without moving from his privacy, Bartleby, in a singularly firm voice, replied, 'I would prefer not to.'" And the story goes on. Every time Bartleby is asked to do something, he replies simply, "I would prefer not to." And it's a little bit baffling because he doesn't quit his job, he doesn't go home, he doesn't abandon his work. He just shows up every day but doesn't do any work, and it drives the lawyer who's in charge a little bit mad. And I really like this story for a number of reasons, but in this case, what I like about this is the attitude of Bartleby is very much like the attitude of the lizards on which we conducted these experiments. And at every turn when we conducted these experiments, they made it very clear that they would prefer not to participate. And they did this by biting us during our experiments. This lizard managed to tie an elaborate knot in his thermocouple cable. This lizard was trying to remove his thermocouple altogether by biting it. This one decided it would rather live in my collaborator Brooke's hair than in its natural environment. This one, I have absolutely no idea what this lizard was doing, but I can tell you that it wasn't thermoregulating in a gradient like we wanted it to. And these lizards would often escape in the lab, and this led us on some crazy chases to recover them. And I want to show this, especially for those of you that are graduate students or postdocs here in the audience. I think it's important to pull back the curtain a little bit, especially if you're working with living organisms, to see some of the chaos and the mess that went into collecting some of these data and the experiments we conducted. In a minute, I'll show you some nice, neat figures of our results that look very great. But I think it's important to know that you're not the only one who's dealing with chaos and unexpected events in trying to collect data from live animals. So with that in mind, let me show you some of the nice clean figures from our experiments. So I'll show you first the results of our very first experiments with the wall lizard, Podarcis moralis. For this, we conducted a transplant experiment, and we brought lizards that lived at low elevation up to extreme high elevation at a lab at the top of a mountain. And you can see one of our results here. So the x-axis here is the 3 time points of our experiment, 1 day, 3 weeks, and 6 weeks. And the y-axis shows their maximum metabolic rate of a lizard. And you can see that across all of the time points of our experiment, that lizards brought up to high elevation under conditions of hypoxia that you see here in the blue circles attained a lower maximum metabolic rate compared to lizards measured at their native elevation that you see there in the red triangle. So this supports the idea, the rather intuitive idea, that if you bring an organism up to high elevation where the oxygen is less available, they'll suffer a reduction in aerobic performance. Any of you that have climbed a set of stairs up at high elevation are very well aware of this. Now, in terms of the long-term implications for fitness, we found that this exposure to hypoxia also affected the body condition of the animals across this time period. So the body condition here is just a scaled measure of body mass to body size. You can see again across the 3 time points of our experiments that after 6 weeks, lizards brought up to conditions of hypoxia had much reduced body condition relative to lizards kept at their native elevation. And this is after only 6 weeks, we see pretty dramatic shifts. So again, this supports our idea that there's long-term implications for this reduction in aerobic capacity. I want to show you one other result from an experiment with the snake, Natrix mora. So this was conducted by a PhD student, Jeremy Suchet. And in this experiment, he incubated the eggs of snakes under different conditions. And you can see here the 4 treatments. He kept eggs at low elevation where oxygen was available and incubated them at cool and warm temperatures. And he transplanted eggs up to extreme high elevation, conditions of hypoxia, and again, incubated them at warm and cool temperatures. And you can see on the y-axis here, the body mass at hatching. So for snakes, the size at which they hatch has important implications for the survival, especially in their first year of life. So we can use that as a fitness proxy. And the takeaway message here is that only under conditions of oxygen limitation and high temperatures do snakes suffer. In other words, if you keep a snake at low elevation where oxygen is available and incubate them at warm temperatures, they can respond to that and they do just fine. At the same time, if you expose them to conditions of oxygen limitation and incubate them at cool temperatures, they can handle it. But it's this combination of reduced oxygen and high temperatures that leads to this dramatic shift in body mass at hatching. So these experiments provided general support for the importance of these temperature-oxygen interactions, but we wanted to conduct a more explicit test of this hierarchical mechanisms of thermal limitation hypothesis. So we conducted this is the final experiment I conducted as part of my postdoc. And for this experiment, we estimated the thermal performance curves of 2 different performance traits for lizards at low and high elevation. And we measured those traits across a range of 7 temperatures. So we measured snakes at their native, close to their native low elevation, the elevation of capture. And then we transplanted them up to a research station at just about 2,900 meters above sea level and exposed them to hypoxia. And this is about 600 meters above their current range limit. So this represents a kind of extreme case of oxygen reduction. Now, here is a view from that mountain lab. This is at the Pic de Midi Observatory. If you're ever in the Central Pyrenees and have a chance to visit, highly recommend it. We conducted a lot of experiments there, and it's a quite magical place. But for this experiment, we didn't have much time to enjoy it. This is one of the most intense periods of data collection I've ever undergone. We had the lab up there to collect the data in a short amount of time. We had 3 respirometry machines going at the same time. You can see our collaborator, S.E. Rogers here, manipulating some of our homemade circuit boards for the respirometry equipment. It was very challenging and very stressful, but I was happy to say I conducted this experiment with 2 good friends of mine from my PhD program, Antonio Cordero and Rory Telemico. And despite all of the stress, we were able to stay good friends and collect some interesting data. So I mentioned that we measured 2 measures of performance. The first measure is sprint speed. This is how quickly a lizard can run under standardized conditions. And this can have important fitness implications in different contexts. So specifically predator avoiding or prey capture. So we measured sprint speed at the 7 temperatures. And importantly, sprint speed is a short burst activity that's overwhelmingly fueled by anaerobic metabolism. So since sprint speed is important, but it's fueled by anaerobic metabolism, we didn't expect to see an impact of this acute hypoxia on sprint speed. We thought lizards brought up to high elevation should be able to sprint just as fast as counterparts at low elevation. Now we think about things like recovery time, that's going to differ, but the actual sprint performance should be unchanged under this acute exposure. For our second measure of performance, I'll turn back to this figure and remind you that aerobic scope for activity is this difference between resting and maximal metabolic rate, and this is important to fuel these long-term processes closely tied to fitness. Now, traditionally, measures of aerobic scope are challenging to obtain in the lab because you have to measure the same individual at all of the temperatures, as well as measure their resting and maximal metabolic rate at 2 different temperatures. So to do this efficiently, we developed a novel method using these remote control robot insects to chase our lizards in our metabolic chambers. So let me show you what that looks like. The idea here is that we have the robot insect inactivated in the chamber. We can measure the resting metabolic rate for a period of time. It's a little tricky for the video to come through on Zoom here. But then after a certain period of time, we activate the remote control and we can chase the lizard, and it results in a really nice elevation of their metabolic rate. And we've since validated this in another similarly sized lizard species. You can see a figure showing some of the results here, and it shows that the maximum metabolic rate attained by this method is actually much higher than that attained by more traditional methods, such as running an animal to exhaustion. So you can see here the time course of one of our measurements. We measure the resting metabolic rate of the unperturbed animal for about 20 minutes. And then at a certain point, we begin activating the remote control robot. And you can see within about 5 or 6 minutes, it elicits this really nice maximal metabolic rate. And then we can take the difference of those 2 lines, and that's our measure of aerobic scope. So now that we have data for these 7, across this range of 7 temperatures for both performance measures, we need to create some thermal performance curves. And to do this, we turn to a mathematical function called the beta function. The beta function allows us to describe the shape of this curve in mathematical terms. And importantly for us, it includes parameters that are readily biologically interpretable and meaningful. So specifically, this curve is parameterized by the maximum performance, WF, the optimal temperature for performance, and the critical thermal maximum. Now, under our hierarchical mechanisms of thermal limitation hypothesis, we predicted that hypoxia exposure would reduce this maximum performance, reduce that optimal temperature for performance, and also reduce, but at the same time, that critical thermal maximum would be unaffected. Now, in this experiment, we didn't actually measure critical thermal maximum, but we had done this in a previous experiment with my good friend and collaborator, Brooke Bodensteiner, you see here. In this experiment, we measured critical thermal maximum of lizards at high and low elevation and found, in fact, that there was no effect. And you can see these results here. CT max on the y-axis here, measured lizards at low and high elevation, and there's no difference. So we're able to use this empirical average from that experiment in calculating thermal performance curves you see here. So we set that to a set temperature of about 41 degrees Celsius. So we created these thermal performance curves. What did we find? Here you can see thermal performance curves for sprint speed. Y-axis shows our sprint performance. X-axis shows our range of temperatures. And you can see those curves are right on top of each other. So exactly as we predicted, there was no effect on this relative reduction in oxygen availability on sprint performance. For our measures of aerobic scope, however, we found a dramatic difference in the shape of these thermal performance curves, just as we predicted. Specifically, that maximum performance was reduced dramatically, indicating that lizards couldn't attain the same they couldn't attain the same maximal metabolic rate, and also the optimal temperature was downward shifted. And this is great. This was the first time in my career, and probably the only time, where empirical results of an experiment matched our theoretical predictions quite nicely. So I like to revel in this figure just a little bit. And so with that in mind, so with this nice result supporting our theoretical expectations, we wanted to take it a step further and describe exactly how these different traits respond to reduction in oxygen availability. And to do that, we created what we called a TOPS, or a temperature oxygen performance surface. And we did this by adding another dimension to the thermal performance curve that describes the relative importance of oxygen availability. And to do this, we modified this beta function. We added a parameter, OF, that describes the fractional oxygen availability. So this is simply the relative amount of oxygen relative to a reference condition, in this case, sea level. And then we added two parameters, gamma and tau, that describe the sensitivity of maximum performance and optimal temperature to this reduction in oxygen availability. And what we hope that in proposing this temperature oxygen performance surface, that in calculating these parameters, they can be compared across different traits within an individual. You can compare the sensitivity of different individuals. You can compare across populations. And ultimately, you could apply this to the intersection of temperature with other limiting abiotic factors. So for example, we could use this same approach to understand the interaction of temperature and, say, salinity for marine organisms, or temperature and rates of parasite infection, for example. So with that in mind, we calculated these TOPS for both sprint speed, you see on the left, and aerobic scope, you can see on the right. And you can see how the shape of these surfaces differ in that aerobic scope is sensitive to oxygen availability, whereas sprint speed is relatively insensitive. And this is borne out by our parameter estimates of gamma here. You can see gamma is about 4 times higher for aerobic scope compared to that for sprint speed. And again, since these are standardized parameters, they can be compared across traits, across individuals, across populations, et cetera, and thus provide a useful tool for expanding this thermal performance curve paradigm to other dimensions of abiotic limitation. So to return to the predictions of the hierarchical mechanisms of thermal limitation hypothesis, we found with this set of experiments that these predictions were in fact supported. So that's very exciting, and it's nice to see the potential to expand this to other organisms, to other situations. And so with that in mind, that's exactly what we did next. And that brings us to our final chapter, but probably not always. So you can see where this is going also. Everything I've described up to this point is our theoretical expectations for what happens when you bring organisms that developed or evolved where oxygen is available up to conditions in higher elevation where oxygen is reduced. That, of course, begs the interesting question, what about organisms that develop or evolve under low oxygen conditions? What happens when you bring them to conditions of relative hyperoxia, for example? And we were wondering the same thing, and so we conducted exactly that experiment. This experiment was with the high elevation endemic lizard Iberolacerta benali. So these are lizards similar in many ways to the wall lizards in terms of their ecology and behavior, but they're restricted to this very small area up in the Pyrenees at high elevation. And for this experiment, we again conducted a transplant experiment, this time taking lizards from their native high elevation and transplanting them to low elevation conditions of relative hyperoxia. And what we found here was a very neat result in exactly the opposite direction that we would have predicted. So specifically, if we measure the preferred body temperatures of lizards, when they're brought to low elevation, they select lower body temperatures despite oxygen being more readily available. And in fact, the effect size here was rather large, almost 3 degrees Celsius cooler, which is a pretty substantial shift in preferred body temperature. We also measured sprint speed, and you see our range of temperatures here on the x-axis for lizards brought to low elevation and kept at their native high elevation. And here you see sprint speed on the y-axis. And at low temperatures, there was no difference in sprint speed. But at the highest temperatures, those lizards brought to conditions of relative hyperoxia actually suffered reduced performance and a slower sprint speed. So let me show these data another way, and you can see that difference pretty dramatically at those high temperatures. So this begs the important question, why? What is going on physiologically that's going to cause these animals to reduce their performance in a number of ways when oxygen is even more available? To understand that, I want to turn us to the mitochondria for just a moment. So a nice recent review paper in Trends in Ecology and Evolution outlines the central importance of mitochondria in ecology and evolutionary studies. This is probably familiar to most of you. The mitochondria, of course, is known as the powerhouse of the cell. It plays a central role in aerobic metabolism, but it doesn't just utilize oxygen to produce ATP. It also balances this oxygen demand with the production of potentially harmful molecules, such as reactive oxygen species. The mitochondria is also interesting because it possesses its own genome. Generally, in vertebrates, this genome codes for 13 proteins that are part of the electron transport chain, so are important for this aerobic metabolism. And this genome can be subject to epigenetic modifications independent of those of the nuclear genome as well, which is something that we know almost nothing about. So turning back to our lizards, I think that exploring the role of mitochondria in oxygen processing under these hyperoxic environments is an interesting place to start to understand this reduction in performance. And to explain that, I want to turn one last time back to our snakes here, the Western terrestrial garter snake, and show you the results from a more recent study in these populations. In this study, we identified a SNP, a single nucleotide polymorphism, in the mitochondrial genome that segregated predictably across the landscape. And you can see on the figure here, 6 populations of the snakes, and you can see the relative proportion of the 2 alleles in the mitochondrial genome. And you can see it's very dichotomous. Populations generally have almost all of one or almost all of the other. Now, this SNP is important because it is in part of a gene that codes for a protein in complex 3 of the electron transport chain. And specifically, the SNP represents a shift from a thymine to a cytosine nucleotide, which results in a change in amino acid from isoleucine to threonine. You can see that here in the protein as part of complex 3, suggesting that it might play an important functional role in the metabolism of these snakes. And I should add that the allele frequencies we see here are unique relative to background nuclear genetic markers and gene flow in the population. So this isn't just a simple product of gene flow, but rather there seems to be evidence that these different alleles are selected for in these different environments. And why might that be? In this snake system, we find distinct life history ecotypes living in these different habitat types. So specifically around the lakeshore, where we find a very high prevalence of one of those SNPs, one of those alleles, we find snakes that grow quickly, attain large sizes, produce large litters, but have short lifespans. In contrast, snakes that live in mountain meadow populations where the environment is cooler and food isn't as readily available, we find the opposite life history traits. Snakes grow slowly, they produce litters only intermittently, but they're relatively much longer lived. So within this context, finding this segregation of these alleles was suggestive that this might play an important role in metabolism. And so we tested this. In one of the populations where we find both alleles, we measured the metabolic rate of snakes. And you can see some of our results here. On the y-axis is the resting metabolic rate. And you can see the results by this cytochrome B SNP, the allele, the mitochondrial allele, and by sex. And you'll notice a really interesting SNP by sex interaction here, where females with one SNP have high metabolic rates that are the same as males with the other allele. So this suggests that there might be some important sex-specific metabolic implications for this allele to segregate across the landscape in the way that we find. So this is very interesting, and this work combined with some other work previously on this snake system led us to propose this kind of generalized framework for understanding these interactions, and specifically how environment and nuclear background interact with variation in the mitochondrial genome to affect oxidative phosphorylation, and how that in turn affects the production of reactive oxygen species and whole organism metabolism, and then how that scales up to variation in life histories and aging phenotypes. So all of that work produced this schematic. And what I'd like to do next is integrate these ideas into some of the lizard systems in these mountainous environments to understand how this might interact with the temperature oxygen, the importance of those temperature oxygen interactions that I showed earlier. And this, of course, brings us back to chapter 2. We think we might understand how with this new schematic, but of course now we need to work through the rest of these steps and test this under new conditions and with different organisms. And I have to say for myself, I'm very thrilled to have a career where this is what I do, where I have the opportunity to work with fantastic people and to develop these ideas and then go out and test them on these wonderful organisms. And with that, I have a few slides here on some of our current work studying invasive populations of these lizards, but I think what I'll do for time is to save these. If anyone's interested, we're also studying these lizards are established in Ohio in the United States, and we're taking a three-part approach to understand how their invasions are successful, looking at thermal biology, morphology, performance relationships, as well as some genomics. I'll save these slides for later. If anyone wants to discuss that, I would be more than happy to. And so with that in mind, let me turn to my acknowledgements. And I'd like to thank a whole bunch of folks and organizations that have been supportive of this work over the years, especially thanks again to everyone here in Montpellier and Perpignan that have been wonderful hosts this week. And I will leave us with one of my favorite poems about a lizard. And I think we have plenty of time for some questions. Thank you very much.
Yes, thank you, Eric, for this very nice talk and stories. I was wondering in the experiment where you play with hypoxia and temperature and the interaction between both, did you ever try to estimate the ROS production level and oxygen stress and things like that? Because when temperature goes high, you can expect that the mitochondria would produce more ROS, and it's a very toxic stress and very damaging. And when you play with the oxygen level, you may also interfere with this.
Yeah, that's a great question. So we did measure some molecules that serve as proxies for reactive oxygen species production in one of our experiments. So we conducted, I don't know if I have slides for this experiment. We conducted a transplant experiment with females, gravid females from low and high elevations. And we transplanted them up to high elevation, and we collected blood samples throughout their reproductive period. And we measured these indicators of reactive oxygen species production. And what we found was actually interesting is that under conditions of hypoxia, later in their reproduction, they actually produced more reactive oxygen species, which is kind of counterintuitive. You think that as oxygen is less available, that they would produce more of these reactive oxygen species. But we in fact found the opposite, and there's support for this in the literature, that this reduction in oxygen availability can disrupt the efficiency of the mitochondria and end up resulting in the greater production. We haven't done that. What would be interesting, I think, to do measure something like that across this range of temperatures, or create, for example, a thermal performance curve for reactive oxygen species, I think would be the next step. So we have this one result in the gravid females that I think is enticing, but I think it would be good to explore this a little more.
So we now have a question from Christophe Gruneau. Christophe, you can turn on your microphone.
Yeah, hi, Eric. Good to see you again. Fantastic talk. Thank you very much.
Thank you.
I was wondering, very nice results about the mitochondria in the L and the M say physiology. I was wondering if you have tried to generate hybrids between the two ecotypes, and if you put the hybrids then in the L condition or in the M conditions, if that would generate a bias. So, I mean, would it just be random distribution of mitochondria, or would the mitochondria, maternal transmission, somehow favor in one environment more the green type and the other more the yellow type?
Interesting. So we have not created hybrids between those two life history ecotypes in the lab. In fact, folks well before my time have tried to get the snakes to breed in captivity, but unsuccessfully. What we have done is a number of experiments where we bring gravid females that are wild collected and then raise the offspring in the lab under different thermal conditions designed to mimic each of those habitats. So it's this kind of cross design between the ecotypic background, so their genetic background, what population they're from, and the temperature at which they're raised. And we have a number of experiments using that design. And we haven't done that in the context of these mitochondrial alleles, but it does show that there is an influence of both this genetic background and immediate environment on immune function, on growth, on metabolic rates, et cetera. And so those adult phenotypes are the product of both these genetic background as well as the environment they experience during development. But it would be interesting to look at exactly what you're saying to better understand it would be interesting to do the hybrid experiment where you can have the mitochondrial genome in a nuclear background that's different from the same ecotype, but we haven't been able to do that. Great question. Thanks, Christophe. Yeah.
Thank you very much, Eric, for a great talk. I have one question. You mentioned you have this hypothesis that at high temperature failure could occur due to organ failure or the macromolecules which are not functioning anymore. Okay. I was also thinking, of course, if you increase temperature, and it would be the same if you lower the oxygen, then you have important changes in the microbiome of these animals. And for sure, they contribute to the physiology of them. I don't know much about lizards, but we know how much it contributes to the physiology of metazoans. So have you looked at this? Do you know any study looking at the changes in microbiome and how it affects its functions, actually?
That's interesting. We haven't done any work with the microbiome. And I don't know over what time scales you would need to measure that for acute experiment.
It could be pretty rapid, I guess.
Yeah, it would be. Yeah. There is, so one study just came out in the past year looking at changes in microbiome of lizards after exposure to acute thermal stress. And they did find that there were shifts in the composition of the microbiome to my knowledge, that work hasn't been extended to the functionality of the microbiome and the long-term implications. But there is a study, at least one that I know of from recent years, showing this immediate effect. So that would be interesting, especially thinking about in the context of the oxygen temperature interaction, if there's a long-term effect in that shift and how that would ultimately perhaps hinder or facilitate their ability to withstand higher temperatures in the future or something like that.
Right.
So yeah.
Okay, thank you.
Yeah, it's a great idea.
Thank you. Really interesting, by the way. In the experiment where you conducted, you put the lizards in the tiny box and then you put like the robots. I was wondering, the tiny box looked a little squeezy for the lizards. Was like, how did you arrive at that particular size? Because they could be stressed with the presence of like a weird thing, you know, moving around them. So it could have resulted in an inflated result for your metabolic rate. So how did you arrive at that?
Yeah, that's a great question. So the size of the metabolic chamber is a function of both the size of the animal and their metabolic rate. So if a chamber is too big, then it becomes more difficult to measure the change in oxygen concentration. You get less sensitivity to that measure. And so those chambers we used were standard size chambers that we had used previously for just resting animals. And what we did was a couple different experiments to validate this. First, we measured oxygen levels in a box with just the robot, no lizard, to make sure that for some reason the moving electronic parts weren't affecting the oxygen availability. We also measured the metabolic rate of lizards in a box with and without the remote control insect, and there was no difference there at all. And so that supported the idea that the presence of that remote control robot, if it wasn't moving, it didn't affect the metabolic rate. It was only when we activated it and moved it around, then the lizards became perturbed. We kind of chased them around and they would run around and sometimes bite. They did not like it at all. And that elevated the metabolic rate. Is that answering your question? Yeah, thank you.
Hello. Maybe you already said it, and I didn't hear it, but when you tested the impact of oxygen limitation, oxygen capacity, of aerobic take rate, but not for the subcellular take rate. And did you test the subcellular take rate for in hypoxia or extreme hypoxia, or you just took it for reference to compare?
Yeah, it's an interesting question, and it's a little bit complicated. Unfortunately, there's not, at least to my knowledge, a specific test for this, exactly which subcellular mechanism is driving that decrease in performance. And in theory, you'd expect that whatever mechanism degrades at the lowest temperature is going to be the one that's important. Assuming that all of those processes are important for an organism to function, you need to pick one that is going to be the kind of limiting factor under different conditions. Now, there's been some work with reptiles showing for example, reaction rates and things like this for enzymes and how they decrease at these high temperatures. And roughly, in some cases, those enzymes functionality will decrease around the same temperature that the organism loses functionality, supporting this idea. We have not done that specifically in these lizards, but I like this. That would be an interesting avenue. What we'd have to do, though, it would be complicated because you'd have to measure a number of different functions, membrane integrity, for example, and then function of these different proteins. But I think starting with proteins involved in the electron transport chain or aerobic metabolism would be a great place to start. Yeah, that's an interesting idea. I want to think about that one more. I like this.
Christophe wanted to ask something else. Christophe, you can hear me now?
I have another question about, of course, mitochondria. When we do microscopy or sequencing or whatsoever, we often see that the number of mitochondria is dramatically different in different cell types, or same species, different strains, and so on. And I was wondering if there is adaptation to whatever, hypoxia or different temperatures. Would it be possible that there's no genetic or epigenetic change, but that the number is changing and that this leads to adaptation? And does it, I assume that doesn't influence your equation, but is this a parameter that change in the equation?
Mm-hmm. Yeah, that would be, let me think about this. So we did do in the experiment that I showed here with the mitochondria, we also measured the cellular oxygen consumption of individual, or not individual, but of different cells, and specifically measured the peripheral blood, the PBMCs, the isolated white blood cells, and we measured the metabolic rate of those cells. And what we did was we normalized our measures by the number of mitochondria, and then from that we could we found differences in the two ecotypes in their cellular metabolic rate. In other words, normalizing for that number indicates that the differences we see at the cellular level mimic those that we see at the organismal level. However, what we didn't test with that is, to answer your question, is about the number of mitochondria in the different cells. And so to do that, yeah, that would be, I think that would be interesting. And I would expect there could be some kind of hormetic effect, exposure to high temperature, exposure to reduced oxygen, to promote the development of these increased levels of mitochondria in something like liver or skeletal muscle. And that would in turn affect the organism's ability to deal with those conditions, although it might not be necessarily in the way that would be intuitive. For example, it might actually be beneficial to have fewer mitochondria after being exposed to high temperature, because then you have potentially reduced metabolic demand under those conditions. So that would be interesting to test. As far as the parameters of the equations, to answer your other question, we thought about this primarily in terms of whole organism performance, but I don't see any reason why you couldn't, for example, measure cellular oxygen consumption or something like this, and use the same approach with that to measure the sensitivity of that oxygen consumption, for example, across temperatures and oxygen availability. So did that answer your question, Christoph?
Yeah, definitely. Thank you.
Okay, thank you.
Hi, I have a question. Thank you for this nice presentation. I'm interested about the last part of your presentation about snakes and SNP.
Okay.
And I was wondering, we saw that there is a kind of relationship between the SNP, then probably the sex, and the performance about oxygen, something like this. And I was wondering if you check the sex ratio among your samples, and could be have an association between the distribution of alleles that you have and the sex, or?
Oh, I see. We didn't find, so in the population, in the wild caught snakes from the populations that we sampled, we didn't find any relationship with sex in that case. Yeah. So, and I don't remember exactly our sample sizes for this, but it was approximately balanced male and female snakes.
Oh, okay.
Yeah.
Okay.
Yeah, that's a great question though, because we would then expect that these effects are very sex specific.
For example, for L3, if it's only females, 14 females in this sampling area, I don't know.
Yeah, no, this is males and females for this year. Yeah, that's a great question.
Thank you.
Yeah.
I also had a little question out of curiosity. I was just wondering, has anyone ever measured quantitative genetic variance through quantitative genetic protocols in terms of how much quantitative genetic variance there is in this metabolic response that selection could act upon?
Actually, let me think if they've done this work. They've done quantitative genetics of some other traits within this system, but I don't think we've looked at, for example, the heritability of oxygen consumption or something like this. There was an experiment that people in the lab did last year or the year before. I wasn't involved with it directly, but the goal was to do some quantitative genetic works with quantitative genetic measures of, I think they were measuring resting metabolic rates, but that work hasn't been published or anything yet. But yeah, but I would be curious about this as well, how much of this is heritable. And it would be interesting, especially in this context, how much is heritable beyond this particular SNP and how that plays out kind of across the landscape here. So, yeah.
Thanks a lot. Okay, so if we don't have any more questions, thanks a lot for giving this very interesting talk, and that's it. Thanks for coming. Thank you.
Merci beaucoup.
Concepts that you